Hello. Okay. Slides. Slides. I do not know why that is being so weird. Essentially, we're going to look at 5 attributes of adaptive immunity. Okay. This is in contrast to innate immunity. It is specific. It is inducible. It can clone itself. It is unresponsive to self. Remember, they had to go to self school to make sure that they didn't bind to the self. And it has memory. Right? This is your acquired. It's going to be acquired over time. It involves active B lymphocytes and T lymphocytes, B and C. We talked about those in the previous video. This should kind of be a review and a little digging deeper and being more specific. 2 types of these immune responses are cell-mediated immune response and antibody immune response. Here is what it looks like.
Host defenses consist of 2 categories, the innate or non-specific defenses and the adaptive or specific defenses.
We talked about this.
The human body has many innate defenses to prevent disease. These include the first-line defenses such as the skin and mucous membranes, phagocytosis of microorganisms, inflammation, and the complement system.
That's innate, last chapter.
The human body has 2 main divisions of adaptive defense, humoral immunity and cell-mediated immunity. Antigen-presenting cells aid in the activation of these defenses. A humoral immune response consists of the activity of B cells and the antibodies they produce. Cell-mediated immunity is a host defense used to fight intracellular pathogens and abnormal body cells, such as cancer cells. The protagonists of cell-mediated immunity are T lymphocytes, which can be divided into 2 groups, helper T cells and cytotoxic T cells. Helper T cells secrete cytokines to activate other cells of the immune system, while cytotoxic T cells attack infected cells directly. The innate and adaptive components of the immune system work together to carry out host defenses. That's what
you're going to have to create for the assignment, for the group assignment. Okay, let's get rid of that real quick. Okay.
Cell-mediated immunity is a host defense used to fight intracellular pathogens and abnormal body cells, such as cancer cells. The protagonists of cell-mediated immunity are T lymphocytes, which can be divided into 2 groups, helper T cells and cytotoxic T cells. Helper T cells secrete cytokines to activate other cells of the immune system, while cytotoxic T cells attack infected cells directly.
Killer T. Okay.
Cell-mediated immunity is a host defense used to fight.
Thank you. Okay. So.
The adaptive immune response is divided into 2 categories, humoral immunity and cell-mediated immunity. A humoral immune response consists of the activity of B cells and the antibodies they produce.
Okay, brief overview of the lymphatic system. Remember, I said these guys are hanging out in the lymphatic system. They're just cruising around looking for stuff. So they're composed, the lymphatic system is composed of lymphatic vessels, lymphatic cells, tissues, organs, and its job is, part of it is to screen the tissue for foreign antigens. So your lymphatic system has B lymphocytes, and they're just cruising for the antigen, right? That's what they're looking for. The lymphatic vessels flow through the lymph nodes, right? This is all like some A&P about your lymph. It has the consistency and composition of blood plasma. So it arises, your blood vessels are kind of leaky and oozy, and they sort of leak osmosis-wise into your lymph. So your white blood cells can patrol that. It looks like this. If you look at it in a book, it looks like a plant lady. That is actually a picture of your lymphatic system. It's super cool. I love the lymphatic system. I think it's often overlooked, but I'm a huge fan. Look at all this awesomeness. So you just have these capillaries, and they're leaky, and they're oozy, and stuff's coming out. And so you don't get edema and explode with like blood plasma all over. It gets caught up in this lymphatic system, and it moves around, and then your white blood cells can be there to patrol it for antigens. You have lymphatic organs, lymph nodes, spleens, tonsils, your thymus, all that good stuff, all in the lymphatic system. Let's talk about the real star of the show, and that is antigens. Antigens, yes. They are the way that your body recognizes a foreign substance and gets ready to attack, right? This is like your killer T's are like, attack, right? What are these antigens? Basically recognized by a three-dimensional region called an epitope on an antigen. It's just like a very specific antigen and receptor situation. Large foreign macromolecules make the best antigens. Food and dust can also contain antigenic particles. It can also be bacterial components. Remember we talked about peptidoglycan is a great antigen because it's not found in anything but bacteria. Okay, here we go. So here's some antigens. Here's the epitopes. So the epitopes are sort of on the antigen so things can find it. So you just have these little antigen presenters and they're like, hey, I got this antigen. There are some types of antigens. There's exogenous antigens. These are toxins and other components of microbial cell walls like the peptidoglycan, membranes, flagella, pili, things that you would find on a microbe. There's endogenous antigens. They're produced by microbes that reproduce inside the body cell, and there's autoantigens derived from the normal cellular process. So here's just some examples of them. See, it's like intracellular. This one is extracellular on the outside. See that little, look at that. It's like a flagellum or something. And here's an autoantigen that comes from normal cells. Okay, preparation for your adaptive immune response, aka your social media platform, aka your histocompatibility complex, or your MHC compound. Your MHC platform that is antigen presenting, like, hey, I ate this thing. Hey, this thing is attacking me. So it's a group of antigens first identified in graft patients when we were trying to graft skin onto other skin. It's important for determining the compatibility of tissue for tissue grafting. Your major histocompatibility antigens are glycoproteins found in the membranes of most cells of vertebrate animals. Hold in position of antigenic epitopes are for presentation to immune cells. So that's really, they're like, hey, I got this thing. We should probably attack it. Preparation for adaptive immune response. Rules of the MHC1. There's MHC class 1 and MHC class 2. We talked about that in my pretty diagram. So pretty. So pretty. Oh, I don't know why it fuzzes it out. It's like it doesn't love my drawing. So class 1, present on all cells except red blood cells. All cells have MHC1. All your nucleated cells have MHC1. Okay, do you see what I just said there? Anyone? All your nucleated cells. What doesn't have a nucleus? Your red blood cells. So it doesn't have MH1 platforms, right? Because it doesn't have a nucleus. So present on all cells except your red blood cells, because your red blood cells do not have a nucleus. MHC2, present only on antigen-presenting cells. This is the macrophages and your dendritic cells. You got this. So they look kind of like this, only much smaller, obviously. And you have these little grooves where your antigen can bind, and this is your MH1, and it can bind there. This is an MH1. On every nucleated cell, it looks exactly the same, but this is really only going to be on antigen-presenting cells. Okay? It looks just like this. They look the same. And in real life, they look like this, like little dendrites. Preparation for an adaptive immune response. So you're going to have to process those antigens, and antigens are processed for MHC proteins to display their epitopes. And it's processed differently if it's going to, process it endogenous or exogenous. But basically it's going to get processed after it's ingested, and then it's going to be presented on an MHC platform. Okay, we will talk about this next time.